Hello everyone, welcome to a brand new video on cellular respiration. This is part 3 of 4 in my series on cellular respiration. In this video we are going to discuss the intermediate reaction and then mainly the citric acid cycle. So we're going to discuss how these two things kind of link together. So what we want to talk about is first our intermediate reaction. We also call this the decarboxylation of pyruvate. In the last video I discussed how pyruvate is produced. Then we're going to discuss the processes of the citric acid cycle. We'll look at how we form citrate. We'll look at the various intermediates, enzymes, and then how these intermediates will become oxaloacetate that then forms citrate to start all over again. We're going to look at what happens at the electron carriers. These will be NADH and FADH2. So nicotinamide adenine dinucleotide and flavin adenine dinucleotide are the two electron carriers used here. So what we're going to start off with, of course, was our whole process that after glycolysis, we produced a 3-carbon compound that was called pyruvate. So what I did in mine is I always just draw the carbon skeletons. And we had 2 3-carbon pyruvate molecules. And this pyruvate is a carboxylic acid, an organic acid, so there will be an OH group on it. And on this carbon here, a double bonded oxygen. So there is a carboxyl group on it. So what we are going to ultimately do is during this intermediate reaction, we are going to decarboxylate it. So the intermediate reaction that we are talking about here is a reaction that is a decarboxylation. Okay, that means we are going to remove the carboxyl group of decarboxylation. And when I remove this carboxyl group, what we're going to do is we are going to form a byproduct, the waste that is important in cellular respiration, CO2. Now, once this also occurs, there's another thing that happens. You're going to have these molecules of NAD+, the nicotinamide adenine dinucleotide electron carriers, and they will come out and form an NADH molecule. Now, also, we have to take this coenzyme or a vitamin A. We're just going to call this CoA. This is what is referred to as the coenzyme A or vitamin A, and it will be added to this process. And at the very end, remember, we produce CO2, and those hydrogens that was involved in the OH, they can be nabbed up, of course. What's going to happen is we now have a 2-carbon acid here, a 2-carbon compound anyway. Now, a 2-carbon organic acid is called acetic acid. But this is not. This does not have the carboxyl group, and instead we're going to have our coenzyme A on it. So we have now made what is called acetyl coenzyme A, or acetyl-CoA. And this is the molecule that will enter the tricarboxylic acid cycle, citric acid cycle, whatever you want to call it. So that's what's going to go in. So we always begin, number 1, with the decarboxylation of pyruvate. And what this does is, 2, we produce this acetate, and then we come in with NADH. So that acetate is formed immediately after decarboxylation. So there is an acetate here that is formed. We then come in and grab those electron carriers, and then we third we add the CoA on to this and form acetyl-CoA. And that is your decarboxylation of pyruvate, the intermediate reaction. Now, this is what goes into the citric acid cycle. The big reason for this is that pyruvate cannot do this because the enzymes that we talk about cannot accept it. Now, I will go in and discuss the enzymes, and we'll put them into here. We're going to start off with the whole citric acid cycle as a whole, and we're going to begin here with our acetyl-CoA. And just for ease, I am now going to put our CoA here. I'm just going to write A, that CoA there. That is the CoA, and just for simplicity, I'm going to be drawing it like that. And what happens is this CoA, there's an enzyme that will add this on, and it needs, remember that many chemical reactions that involve enzymes require some sort of cofactor, whether it is an inorganic substance or an organic substance. An organic substance that is a cofactor is referred to as a coenzyme or a vitamin. So we're going to begin here actually with the end product. I'm going to begin with the end here. And I'm going to have a 1, 2, 3, 4 carbon substance here. And it's very hard to write its name. It's oxaloacetate. Oxaloacetate. Now, this oxaloacetate molecule will come in and accept this vitamin A, basically vinegar with vitamin A on it, acetylcoenzyme A, and the CoA will go off. The CoA, the vitamin A, will go off and be released. And this, I'm going to show you guys, is what we're going to do. We're going to take this original skeleton here, this 1, 2, 3, the skeleton of oxaloacetate, and showing that we have it. Now, from here on out, what I'm going to do is I'm going to take these two carbons and I'm going to not color them in black. Instead, I am just going to attach them here, and I'm just going to make it a circle so we can see, kind of help keep up with it. Okay, and now we have made citrate. Now, this is why it is called the citric acid cycle. Citrate or citric acid is produced from this. So what happens is we take acetyl coenzyme A, we fixate that to an oxaloacetate to form citrate in the very first step of the citric acid cycle. Now, what we will continue on to do in the citric acid cycle here is we're basically going to have involved water being added and removed. So we have hydrolysis and dehydration reactions occurring here. So we can shuffle some bonds around. So there is dehydration and hydrolysis reactions. And one of those things that's going to happen is we are going to lose, we're going to modify this thing around. We are going to basically create a modified structure. It will look the same to us, but if you were looking at an overall chemical structure, we're just basically going to shift it around a little bit and form an isomer. Now, you cannot see this in my drawing, but if you look at a biochemical text where it actually does show these, you will see these changes happening. And what we're going to be having here now is a molecule called isocitrate. Now, don't worry, I will come back and talk about the enzymes that do this in a minute. Then what we're going to do with our isocitrate, we're going to come in with this. We are going to take an NAD plus molecule, and we're going to make an NADH. Remember, these are like poker chips who can be cashed in later for money. And also in this process, we lose some carbon dioxide. That means one of the carbons that I have on my black skeleton here is going to have to be removed. So we're going to take that 1, 2, 3, and I'm just going to remove that side one. And then let's put 2 of our acetates up there. And we have now formed a molecule called alpha ketoglutarate. Alpha ketoglutarate. Now this alpha ketoglutarate, as it goes in, we now have another NAD+ molecule here. And we also have more coenzyme A needed. Okay? And what's going to happen? Let me kind of draw the arrow continuing on here. This NAD plus forms some NADHs, or another NADH, just one more. So we now have another NADH formed. And this coenzyme A comes in, and what we are going to produce now is a molecule that is somewhat different to alpha ketoglutarate. It's going to have also having lost one of its carbons here. I did not show that carbon change there, but we do remove one. Let me put the CoA here. And then what we're going to do is just, let me erase that a little bit too. And actually that'll help me. Okay, so I just want to make sure that I had that exactly the way I wanted it. Okay, and I like drawing these out as non-filled in balls. So this acetyl-CoA comes in. It's going to be very hard to write this in. This molecule here is called succinyl-CoA, and I'm going to try to write that in right here. Succinyl-CoA. And what is going to happen, just like before, as we round out this reaction, you are going to remove the CoA. Why is it there? The enzyme that reacts needs that. And actually, before I get myself into some trouble, let me change this a little bit. There we go. I could see I was going to paint myself in a corner if I wasn't careful. So we're going to lose that CoA. Students oftentimes ask me, why do you do that? Well, just remember that vitamins are needed for certain chemical reactions. So if we don't remove it, we're not going to actually be able to do the chemistry with that enzyme. Now, we're going to have some ADP, adenosine diphosphate. It's going to come in here, and we're going to form some ATPs. Remember, this is substrate level phosphorylation. There's a kinase that is involved in that reaction. Now, what we basically have is something that is not going to be like anything left from the original chemistry. So let's draw 1, 2, 3, 4 here. Let me try to really make these stand out. Now we had succinyl-CoA because we had 1, 2, 3, 4 carbon. 1, 2, 3, 4 carbon. And this is succinate. Now I do have mnemonics to help you guys learn these. Then with succinate, we are going to come in with our flavin adenine dinucleotide, FAD. Now, FAD comes in really late in the game, and that mostly is because it doesn't have this positive charge, so it doesn't have as great of affinity. So it's easier when we have broken it down a little bit and degraded this larger molecule down. It's got less electron affinity. And it's going to come in, and it's going to form an FADH2 molecule. And then we're going to have a 1, 2, 3, 4 carbon substance, 1, 2, 3, 4 carbon substance called fumarate. Now, I'm going to write fumarate inside here. Now, this fumarate molecule, as it comes around, we are going to add water to this, which means some hydrolysis can happen. Now, what we're basically going to do is we're going to take this fumarate, we're going to add water to it, Then basically we're going to make a molecule that is called malleate. We're going to make some malleate here. And what we want to do when we form this malleate is also, lastly, to form oxaloacetate, you need NAD+, forming NADH molecules, NADH molecules. Okay, very hard to do the citric acid cycle on this tablet the way that I did it, but we did get through it. What I want to do is begin to fill in some of these enzymes and talk about what is actually happening with some of these molecules that we talked about. Now, first off, let's help you guys remember some of these in order, some of the intermediates that we just did. Now, there are two ways to remember it. One of the easy ones that is common out there is citrate is Krebs starting substrate for making oxaloacetate. Okay, citrate is Krebs starting substrate for making oxaloacetate. Now, why that is a good one to use is, well, number one, citrate is isocitrate. Now, the hardest thing about this one is Krebs is alpha ketoglutarate, starting substrate. Now, one of the things that is kind of harder about this one is you have two S's. You have first, now a good way to remember this is A is the first letter in the alphabet. So we have a succinyl CoA, if you remember that A is the first letter in the alphabet. And then we have succinate, 4 fumarate, M malleate, and then oxaloacetate, which can make a lot of these easy to remember. Okay, so another one that I use, I do have this in my lecture notes when I teach it. Luckily, since it's college, you can use some slightly PG-13, NC-17 rated mnemonics. And one of the ones I use is, can I keep selling sex for money, officer? Is another one that I use in my classes. Now, what we then have to do is understand there's many enzymes involved here. Now, it's very hard to write the enzymes in, and you can easily do this because there are enzymes that we have that go through and add these things in. It's very hard to actually draw it out in this. So what I'm going to do is list them, and very easy to help you remember them in order. They go with this. Like to form citrate, you need an enzyme that synthesizes citrate. Very easy to remember that is that it's citrate synthase. So now, a lot of mine either deal with drugs and alcohol, especially alcohol, because it's college students, and we know college students like to drink. So, can Anthony drink down 7 drinks for death? 4 being a contraction, kind of abbreviation of for death. So can Anthony drink down 7 drinks for death? So before he dies, can he get 7 drinks in before he dies of alcohol poisoning? All right. Now, that is, so can Anthony, so we have citrate synthase, to synthesize citrate. Now, Anthony, our next one is called aconitase. Then pretty easy because we saw that we had isocitrate we were working on. So isocitrate dehydrogenase. Dehydrogenases, remember, are involved in transferring electrons to electron carriers. We then have alpha ketoglutarate dehydrogenases. Now, this is where you have to remember the D's here are standing for dehydrogenases. Okay, so any D that is in this is dehydrogenases. And oftentimes to remember, it's just the enzyme that is dehydrogenating that preceding molecule. Now, we have succinic thiokinase. Succinic thiokinase. Then we have succinic or succinate dehydrogenase. Now, just remember that any of the D's in my mnemonics are standing for a dehydrogenase. Fumarase. And then malleate dehydrogenase. Now, these are all, remember, we talk about dehydrogenases, dehydrogenate. Now, we do know that these electron carriers, such as let's say NADH, is carrying with it electrons. And we know that they're there, so where does these electrons go? Well, these electrons that this molecule picks up ultimately goes to our next video, the electron transport chain, to be utilized. Remember, these are like poker chips that are worth something, but you can cash them in for money. So guys, this actually does conclude my last video on the preceding preparations for the electron transport chain. Electron transport chain is where the money is. That's where we're going to make all of our ATP, and that's going to become the last video. So I hope this video helped you. If you find my mnemonics useful, if you have something you would like to share, or if you have future topics you'd like to see me do, or anything else, please let me know. Don't forget to comment, like, and subscribe. And as always, thank you guys for watching, and I appreciate every one of your views. Thank you. Have a great day.